This is an A-level presentation looking at active transport and co-transport. In this presentation, we're going to go through the concept of active transport. We're going to make some comparisons between active transport and diffusion, and we're going to look onto co-transport and specifically co-transport in relation to the absorption of glucose. So start us off, have a think back. What can you remember from GCSE about the concept of active transport? Hopefully you will have had a couple of thoughts in your head which go along the lines of energy is required and its movement from a lower concentration to a higher concentration. You may also be aware that it involves the use of proteins. So active transport is the net movement of molecules from a region of lower concentration to a region of higher concentration against a concentration gradient, and this requires ATP and the use of carrier proteins. And if you're not sure what carrier proteins are, go and have a look at the A-level presentation on the structure of the cell surface membrane. Active transport requires ATP, and the ATP is used in two ways. The first way is that it can directly move molecules across, and the second one is that it can move molecules across using a concentration gradient that's been set up by co-transport. So co-transport is a specialized type of diffusion, but it's dependent on the active transport of a particular ion, which then causes the co-transport of another substance, and the example we're going to look at is glucose. Again, ATP works in conjunction with carrier proteins and can move substances across the cell surface membrane. There are different ways in which we can adapt surfaces to maximize the rate of diffusion, and in terms of active transport there are several factors. Pause the video for a moment, have a think about what the images may represent, and when you're ready, replay the video. So we can increase or decrease the rate of active transport by increasing the number of proteins in the membrane, by changing the concentration gradient, by reducing the thickness of the exchange surface, by increasing the surface area over which diffusion takes place, and of course by changing the temperature, and that relates to the concept of collision theory. Comparing diffusion and active transport, they're essentially opposites. One involves a net movement from a high to low concentration, where the other is a net movement from low to high. In the active transport, we have carrier proteins required and the necessity for ATP, whereas diffusion, no proteins are required and there's no need for ATP. Direct active transport is essentially when you have a cell surface membrane, a molecule hits that carrier protein, and the ATP is used to move the shape of the protein, and by moving the shape, the molecule is released on the other side of the cell surface membrane. And we're essentially pumping substances from one side to another, and there's a number of examples in that. The sodium-potassium pump arises in several parts of the biology course. Now, co-transport is a very specialized type of diffusion, and it is dependent on active transport. In the diagram, what we have is at the top we have the lumen of the ileum, so the small intestine, and that's the inside of the small intestine. We then have the epithelial cells that make the lining of the ileum. So they're the actual outer layer that make up the ileum wall. And then surrounding that we have, of course, the blood capillaries. And we have the movement of substances, and the substances we're looking at are glucose, sodium ions, and potassium ions. And the entire mechanism starts, as we said, with co-transport, is dependent on active transport of a substance. So we have to start with the active transport of the sodium ions. By actively transporting the sodium out, we are decreasing the concentration of sodium inside the epithelial cells of the ileum. What that means is we get a concentration gradient between the lumen of the ileum and the epithelial cells that results in the sodium moving into the epithelial cells. As the sodium moves into the epithelial cells, it brings with it a glucose molecule because you use a co-transport protein. Both are moved across at the same time. As they move across, we are increasing the concentration of glucose inside the epithelial cell, and we're increasing the concentration of sodium. Once again, the sodium will be pumped out, whereas the glucose can move through another protein and it will diffuse out into the blood capillary. But because the diffusion requires a protein to be present, we refer to that as facilitated diffusion. It's not simple diffusion, it's facilitated diffusion because if the proteins weren't there, the glucose molecule will never make it into the blood. And then we can start the whole process over again, and in this way, because the glucose will only move through the co-transport molecule when the sodium ion's there, and we're constantly creating a sodium ion concentration gradient, that means that the sodium ions are constantly moving into the cells, we will always have glucose moving into the epithelial cells regardless of the concentration of the glucose. And then all we'll have is, depending on the glucose levels in the blood, the rate of movement from the epithelial cells into the blood by facilitated diffusion will vary.